Hello, in this video we're going to introduce how to use PCR, simulate PCR in MCDS. So this is a typical PCR node. You click the PCR node and you can see the details of the operation property. You can find the primer name, the primer sequence, and you can type a primer. So for example, you can select the primer from here, like MPF, RPF. If you select the primer sequence will change here. And you can tap MPF, press enter, or you tap RPF, press enter. But anyway, we're going to show how to design the primer. So by click this PR button, there will be a PCR tab show in the dashboard. So here we want design primer for RFP. So we select RFP and set the minimal melting temperature 62. So we click here, we get the forward sequence, the forward primer. And we click here, we got the reverse primer, and we'll give its name RFPF. And by click pair, it goes back to the operation property and do apply. It will generate the primer. Generated the products. And there's another search mode for the primer. For example, if you want search verification primer for this part, you can click find, select this region first, select this feature, or select this fragment, and click find. So this will find primers within the 250 base pair. So if you want to clone something, but especially when the fragment is large and you are not sure and a primer arbitrarily designed would work properly, you better select the whole fragment you want to clone and use the search function to search the best primer for you. And by click this arrow, you can see a list of search primer which generates which has a max hairpin, max cross diameter parameters here, and tells the distance of the primer, how far the primer is from the end of the selection. And in this list you can select the user pair to select the pair primer, or you can use the forward primer, or just use the reverse primer. It will change the primer sequence here. So now let's go back to the PCR panel, the PCR node. So after design this primer, we find that the PCR products are not just one. That's because the PCR algorithm in MCDS actually simulates the process of the real PCR. It generates single strand intermediates and try to anneal them together. So that means with this plasmid, if your PCR enzyme can generate as long as 8 or 14 kilobase pair, you will really get those products. And for example, this is another example which has two primer bonding sets for RFPR. So if we use this one as template and we tap RFPF and RFPR and we apply, you will get two products. That's because you have two primer bonding sites. So that means MCDS actually simulate the PCR process. And in PCR node, you can copy both primers. So for example, if you select the PCR node and press Ctrl C, by default, you will copy both PCR primers. So you can paste in a notepad. You can see the primer here. And in the PCR sequence, we can see this greater than mark. This greater than mark actually means the part after the greater than mark is the bonding size, and before the greater than is the attached sequence. So for example, you can put BAM-H1 here, and it will give you BAM-H1 sequence directly there. And you can tap equal R1 here, you get equal R1 sequence. And then you press PR, you see the PCR tab. And in the PCR tab, by default, if the MCDS can search the bonding sites, it will show the PCR region in red. And here, we show how the bonding sequence works. So for example, when we design primer in the sequence viewer and click in the sequence viewer, you actually only change the bonding sites. You won't change the attached sequence. So this is make it much easier for you to change the bonding sites without modifying or redesigning the attached sequence. So you can also only pass this forward sequence to the operation property. For example, we pass the forward sequence as R primer to the operation property. So we click R. So RPR goes here. And we can pass the RPR to the forward one. So we click this F. So RPR goes to the forward primer. So this pair will pass the whole primer, the whole pair of primer to the operation property. So click apply, you get primers. But because we have another node, 11, which also use RPF and RPR, but without the attached sequence. So MCDS generated the warning that you have primers with different sequence but have the same name. So we better change the name RFPFB, RFPRE. So if you change the name, there will be no warning. So that will help you avoid conflicts of primer names. And to avoid a pair of primer to produce multiple fragments, especially when we want to do design for given assembly, we only want one product. So we probably can digest this plasmid and just get one fragment of it. So for example, like here, we digest with equal 1 and the SPE1, and only get the shorter fragment, and we copy this primer pair and create a new pair, new PCR node, and paste. Press Ctrl V to paste the primer pair. So that means you can here Ctrl C copy the pair of primer and paste in the PCR node. So now, if you want to design a primer for restriction enzymes like this, you put BAM-H1 equal 1 size. You probably add some protection sequence. For example, we put 3A here, you can see the hairpin and the dimers are not increasing. That means those A doesn't make your or decrease the quality of your primer pair. And you can also add a T here, probably no, C, yep. So this won't decrease the quality of your primer. So you get a powerful primer with resolution size and protection sequence. And for the design of Gibson assembly, we have a Gibson assembly designing node. So for example, we want to put 3 fragments together like this. Here we better change the RPF name because Gibson assembly design will modify the sequence by putting attach or annealing sequence. So we put RPR3 here. You will get the warning because you have primer pair with different name but same sequence. So it doesn't matter. So we select all the 3 fragments. We want to do Gibson for all the 3. So we click the Gibson design and click apply. So we back to this node. The Gibson design has attached the annealing sequence to this pair of primer. So now it will generate a plasmid from those 3 fragments. You can also just design the Gibson assembly for only 1 pair of primer. For example, in the previous example of the Gibson assembly, it also attached the sequence to this pair of primer for PAC18 replication arranging because they are all PCR node. If you want to just design a Gibson assembly primer for digestion fragments, for example, we digest it with equal R1, we get these fragments. Let's remove the attached sequence. Click apply. There's a warning, it doesn't matter. So we do Gibson assembly, and in this case, you can see that the node 15 is just a digestion result, a digestion fragment. So the Gibson assembly designer cannot change it. It can only change the PCR node. So just click apply. It will attach proper annealing sequence to the PCR node. Then you get the RFP inserted into this PAC18 vector. You can also design primers for verification. For example, after the Gibson assembly, we want to verify if the PRP fragments goes into the PAC18. So we want to design a primer, press Ctrl R to get a PCR node, and press PR. We get the tab. So we want to make sure the RP is connected into this plasmid. So we select this region. We want a pair of primer to confirm they are joined together. So we select this one and press find. So we find a pair of primer within the 250. So here you can see the attached sequence. The bonding sequence. And those are the hairpin dimers and cross dimers can be found in those 2 pair in this pair of primer. You can see that there's no hairpin, no dimer, no cross dimers. So that means they are a good pair for primer. So we can assign name verify F, verify R, and pass this pair of primer to the operation property and apply. So we get the verification products 356 base pair. So we can also look at the detail of the primer with the attached sequence. For example, this one. We click the PR and check this panel. So you can see the highest, the hairpin with the highest Gibson energy change for the forward primer, reverse primer, and the diameter and the cross diameter for them. And can see possible product length 0, 1, and mismatching here. And see here, this is very important. The A means the melting temperature of the whole sequence, and the B means the melting temperature of only the bonding sequence, and the 19 is the length of the bonding sequence, and the 29 is the length of the overall sequence. So we're back to the property panel. You can see 29, 31 here. So that's the information in the PCR tab. And we can also design primers for performing lambda red knockout. For example, this is a E. coli genomic fragment which has TOR-T and the TOR-S gene. And so we want to design a pair of primer. Use a template PKD3. So for example, we create a PCR node and click PR and amplify this chlorophenical resistant gene. So we design the primer here for the forward, and here for the reverse. And we tap the primer name, pass the pair of primer to here, to the operation pair property. We generate the product. So in order to design lambda red knockout primer, you know that you usually need 35 base pair, nucleotides, homologous fragment. So you can actually copy from here, select the 35 from the sequence viewer, copy, and paste it here, and again back to this one. You can see the TOR-T feature here. We select another 35, see here is 35, we copy it, and this time is reverse complemented. We click reverse complement and paste it here. So we generated a pair of primer for lambda red knockout. So we now do the recombination. We select both nodes and do recombination, and here we select lambda red recombination. So we click apply. So we can generate 3 fragments here in the products. So we want to find the one with chlorophenical resistance here, and just use gel separation, because the gel separation can separate them with the length. The 1.1 kilobase pair one is still the fragments, and this one is the fragment, the genome fragment replaced by chlorophenical. An alternative way to design the primer for lambda red knockout or other recombination is to generate a fragment and use the Gibson assembly. For example, we copy the sequence before T, whatever the length it is, maybe a bit longer, and create a free designer, apply, and again we copy the sequence after it, maybe a bit longer. Yep. So back to the free designer, we put this one before it. So we select those two nodes and use the Gibson Assembly Designer for it. In Gibson Assembly Designer, you can specify you want at least 35 or 36. This time we use 36 for the annealing. Then we click apply, so you can see that you got the 36 nucleotides in each of the primer for the lambda right recombination. So back to the lambda right, we select those two fragments, do lambda right recombination, apply, screen for paraphenical, and get the bigger one. Yes, so it's still working. Alright, so that's basically how to use the PCR node and the Gibson Assembly Designer to design your PCR primers in MCDS.